Good afternoon. My presentation will focus on CRISPR-Cas9 technology, pre-implantation genetic diagnostic technology, and how they both relate to designer babies and the possibilities of them in the future. For the past few decades, especially in the late 80s and early 90s, the idea of genetic modification and genome editing to create a, quote, superhuman has occupied the thoughts of many, as per an example in the movie Gattaca. However, the technology portrayed by Hollywood is just not obtainable. It's too far off and way too expensive to logically procure. However, there are some examples that have brought this idea a little bit closer to home. Although these technologies are not quite what Hollywood portrays as giving super strength or the ability to fly, they are too incredible. CRISPR-Cas9 technologies, and in combination with PGD, or pre-implantation genetic diagnosis, have both combined together to help form a new technology and to create a more realistic superhuman, if you will, referred to as a designer baby. As you see in the second bullet on the slide, both technologies, CRISPR-Cas9 and pre-implantation genetic diagnostics, both combine to a common term. They both focus on genetic modification to give an advantage. However, they are not identically. As you see from the third bullet, CRISPR-Cas9 focuses on gene splicing, which is going in and physically editing the gene, while pre-implantation genetic diagnostics is merely a screen for a said disease. It doesn't actually go in and fix it. It merely raises a red flag. The first example, CRISPR-Cas9 technology, has only rarely become available recently. However, this breakthrough technology has finally opened up the option that we've sought for so long. CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats and CRISPR-associated proteins. Essentially, how this CRISPR technology works is the scientists go in to the genetic sequence, to DNA itself, and they figure out through a highly scientific process, which I have not been able to wrap my mind around yet, they essentially find out which gene it is that causes a specific disease or is causing a specific condition. Then they go into the DNA itself with some other highly skilled scientific process, and they physically edit the gene. So, say you have a C, cytosine that's causing a certain disease. They will go in and they'll edit the gene and change it to another base that will eliminate the possibility of that disease. While this is far more recent and therefore much expensive than PGD, it is far more local and far more versatile, with many major implications for the medical field. One of the biggest questions I had while doing my research was what you see in the second bullet down, which is CRISPR-associated proteins. I had a lot of trouble finding out just exactly what a Cas protein was. And it finally took me, after reading about 15 or 20 articles, I came upon an article which finally described what it was. Essentially, a Cas protein is any protein that is associated with the CRISPR process. The following is a quote taken from the article which finally helped me understand this process. Quote, "4 CRISPR-associated (Cas) protein families designated Cas1 to Cas4 are strictly associated with CRISPR elements and always occur near a repeat cluster. Some spacers originate from mobile genetic elements and are thought to confer, quote, immunity, unquote, against the elements that harbor these sequences." After trying to wrap my head around this for quite a long time, I finally boiled it down to the basics. Essentially, what this says is that these Cas proteins, labeled 1 through 4, occur with the CRISPR process. Furthermore, it states that these elements of spacer DNA protect the CRISPR repeats from any harm while they do their job, essentially guarding them. And one thing to keep in mind is that spacer DNA is a non-coding region of the DNA. Essentially, it's just a placeholder which is guarding the other CRISPR parts that are doing the work. They're helping protect these CRISPR sequences. As you see in the slide, a designer baby is a baby genetically selected in vitro for specially selected traits, which can vary from lower disease risk to gender selection. In other words, what a designer baby is it's a baby whose parents are trying to influence certain qualities of the baby, from hair color to eye color to making the baby slightly more resistant to diseases, essentially giving the child a better start or better future for life, which, after all, is what many parents would want, or really any parent would want. There is an ongoing ethical debate about how designer babies can be used and if they're even ethical to be used in society. However, we'll cover that later. As previously stated, the other technology behind the designer baby concept is far different than that of CRISPR. This technology was first introduced in the United Kingdom in the 1980s in humans, although it's actually been around since the 60s, just not in regards to humans. Essentially, the process of pre-implantation genetic diagnosis is this. An embryo is selected and then screened for certain undesirable traits, such as diabetes or Huntington's disease or possibility of cancer. If the test comes back positive for any of these diseases and the parents decide that they don't want the baby to have to go through that, this test will raise a red flag that will see the disease, and then the parents can go in and select another embryo and test it and see if that embryo is at risk for said defects. PGD merely screens the embryo for the disease, letting the parents know about the possibility of future medical defects. If one or both of the parents carry a gene for a known disorder, the embryo is tested for that defect. The actual screening, the physical screening of this is considered to be the genetic diagnosis. This is one part of this that I personally struggled with understanding. For quite a while doing the research, I was under the impression that there was some magical technology, like CRISPR, that would modify the embryo. However, upon doing further research, I realized that it was just a screening, and CRISPR is the only thing that would actually modify the gene. When I was doing my research, I commonly came across one myth that seemed to be circulating around the internet and even through doctors who this was their field of study. It's a myth that I saw so often that I saw fit to include in this presentation and to dispel any rumors about this. That way everybody will be completely clear. The following is a quote from a medical journal. Quote, "The most persistent misconception about the medical process that produces so-called designer babies, called pre-implantation genetic diagnosis, is that scientists create new embryos that possess certain characteristics. However, PGD is a process of selection, not creation. First off, couples are always undergoing in vitro fertilization before they can choose PGD. This requires the doctor to create embryos by manually combining an egg and sperm in a laboratory dish. This is the only way an embryo is created. Doctors cannot alter the characteristics of the embryo before or after it is fertilized. A clinician creates a number of embryos, then lets them develop for about 3 days before looking at the genes and assessing them with this test. This is where the word selection comes in. If a couple learns that an embryo tests positive for Huntington's disease or some other disease, they may choose to implant a different one. As far as applications for PGD goes, there really only is one application, and that is the screening test. This is because this technology is somewhat limited in its availability to do much for the baby. Like I've previously stated, it doesn't edit any of the genes, it doesn't edit anything within the embryo. What it does is it goes into the embryo and it looks at the contents and then evaluates the embryo and decides whether or not this embryo has potential for future medical implications. If it does, it will raise a red flag. In contrast, the application for CRISPR-Cas9 technology is far more versatile and widespread. There are three main applications: agriculture, disease modeling, and gene therapy. The fourth one I listed, I just found out that in China they are testing CRISPR-Cas9 as a possible cure for cancer. However, this has not been confirmed. As far as agriculture is concerned, CRISPR technology can be used to improve qualities inside the plant, like disease resistance and drought tolerance. And instead of having to rely upon common methods, such as genetic variation and regular fertilization, scientists can go into the plant, and once they've identified a certain gene that is susceptible, they will go in and target said gene, therefore making the plant much more efficient and reliable. Disease modeling, by definition, is an animal or cell displaying all or some of the pathological processes that are observed in the actual human or animal disease. In order to study how diseases affect humans, and in order to explore possible treatments without endangering human life, doctors perform research on these cells, which are similar to human cells. However, a big challenge that doctors face is that while the cells are similar, they aren't identical, which still leaves room for error. By engaging CRISPR technology, doctors can selectively modify these cells so that they are the closest to human cells as possible, thus giving the most accurate data and most accurate results. The last definite application for CRISPR technology is gene therapy, which is the main use for CRISPR. Gene therapy is the transplantation of normal genes into cells in the place of missing or defective ones in order to correct genetic disorders. This is the exact process that goes on when doctors try and employ CRISPR technology to rid an individual or a designer baby for genetic disease, such as Huntington's. As I previously mentioned, in China they are currently testing CRISPR technologies in lieu of treatments such as chemotherapy, as it's far less invasive and far less harmful. As I've maintained throughout this entire presentation, designer babies essentially combine these two technologies into one whole experiment. When people commonly hear the term designer babies and they think of genetic engineering or genetic modification, usually the first thing that comes into their mind is an example of Spider-Man, or an example of some superhero that has a modification that allows them to climb walls or fly or have some crazy ability like super strength and the ability to lift up a car. However, as far as the current technology and the current society is concerned, this simply isn't true. Designer babies, as far as that goes, are pretty simple. They culminate CRISPR technology, which is gene editing, the more complicated process, with the ability to screen for diseases, which is represented by pre-implantation genetic diagnostics. They are a culmination of both of these experiments. They're able to roll them all into one. And they are not some genetic mutated freak show like I first thought when I started doing my research. As far as ethics goes, it's currently a controversy. As you can see in the slides, there are quite a few good reasons as to why we should have designer babies. They can help increase the lifespan. They'd be able to give people a better head start, and possibly they could help rid current diseases that cause so much heartache and trauma in our society. such as Huntington's disease, Alzheimer's, and possibly even cancer if the treatment does go through. It could also eliminate diseases such as obesity, anemia, diabetes, just to name a few. While having this technology would be awesome, and for many people it would be life-saving technology, and I do fully support this as my own opinion, there is the other side to the argument in which there are some negative impacts that could potentially destroy society. One of the biggest ethical concerns is the gap in society, especially when it comes to the rich having just another advantage. Right now, this technology is rather expensive if you can afford it, and that could create a diverse gap in society. The rich have the money and have the means to have this genetic modification and would set them apart. It would just create further animosity and resentment between the classes. And perhaps the most disastrous negative of this potential technology is what you see in the last bullet. Now, the current technology does change the genes a little bit. It physically changes the DNA. And one of the things that worries scientists is, some genes have multiple functions. And so if you change that one letter and you're changing the sequence, who knows what else you could be changing? You could be changing a critical infrastructure to the human body, and you're not going to know until that whole thing goes awry, and then potentially you're going to have a death on your hand. And for something that is experimental as it is right now, that's a pretty big risk. That's a pretty big unknown to take. I hope you've enjoyed my presentation on designer babies, CRISPR technology, and pre-implantation genetic diagnosis. On the following 2 slides, you'll find my references. Thank you, and have a nice day.